Our next talk is going to be about remote diagnostics, and I'm going to bring in a couple of speakers from Fera Science Limited. So I don't know who's going first. So Ashley, excellent. Over to you.
Hi everyone. Today I'm going to be talking about work we did on PathSafe Workstream 3A, which was on rapid diagnostic technologies for foodborne pathogens. So the workstream was split into three main work packages. The first focused on a literature review of on-site technologies for foodborne pathogens, and alongside this, a technology readiness level framework so we could assess where these technologies were in their readiness for deployment. And these were put together into a database of the technologies with their assessed technology readiness levels. Alongside this, in Work Package 2, we engaged with stakeholders to see where on-site technologies could be best placed and provide most benefits. And these both fed into Work Package 3, where we selected two technologies for pilot study. And this is going to be the focus of the presentation. So just briefly, our technology readiness level framework sort of described the steps that need to be taken for deployment of an on-site technology, from concept feasibility all the way to full diagnostic performance measured in the field with end users. And if you're interested more in the results of this part of the project, we have a poster on it. So we engaged with both strategic and operational end users to see where these technologies could provide benefits and where they might be feasibly deployed, and also to get ideas on what would be expected from the technologies in terms of speed to result, ease of use, performance, and cost. So these were some of the needs and opportunities identified during the end user study. So for statutory testing, for example, pen-side testing or product testing at points of entry such as ports, and then for non-statutory or additional testing, customer assurance of safety, for example, in shellfish, hygiene testing at production facilities, and help with production decisions such as the safety of irrigation water. Test requirements were obviously scenario specific, but some of the things which were mentioned as desirable were being able to tell if pathogens were viable, providing some level of quantification to monitor trends. High sensitivity was vital for a lot of the scenarios, and then cost, speed, and ease of use were also desirable. So we put together our results from Work Package 1 and 2 to decide on technologies to take forward to pilot study. So we prioritised technologies at sort of mid-technology readiness levels so we could potentially progress to using them with end users, and then try to match these shortlisted technologies where they could provide benefits for a certain scenario. And the two we chose to take forward to pilot study were monitoring E. coli in irrigation water for fresh produce using portable real-time PCR, and detection of salmonella in high-risk foods of non-animal origin at ports using LAMP. So first, portable real-time PCR was chosen to go forward with this scenario because it was the technology which had the most potential to offer quantitative results to monitor trends. And during our literature review, we assessed multiple portable real-time PCR platforms, but the one we chose to go forward with was the Franklin by Biomeme, mainly because it had a commercially available kit for detection of E. coli, but with no published validation data. And also the workflow looked easy to use and rapid, which would be important for end users. It also had been assessed in the field for COVID-19 testing at hospitals and African swine fever virus testing at farms with good performance data. So the first step in our pilot study, we assessed multiple assays to combine with the portable real-time PCR platform, including their commercial kit and also two assays from the literature. So we assessed that analytical specificity using a panel of target bacteria and closely related non-target bacteria, and all tests showed good specificity with no cross-reactions. And for analytical sensitivity, we used dilution series of E. coli DNA. And again, all the tests performed quite equivalently. Therefore, we chose to go forward with the commercial kit because of its obvious benefits for ease of use for the end users. We then went on to develop on-site water filtration and DNA extraction methods. So the two methods we trialled were filtration with a filter funnel and a vacuum hand pump, and then syringe filtration. And the first method is the one we eventually chose just because it was easier to use. And then when testing with spiked samples, it also gave lower CT values, so suggested higher sensitivity. We then compared an equipment-free DNA extraction method using a cartridge just to a QIAGEN gold standard lab extraction method. And again, it performed quite well, only giving CT values about 1 cycle higher with spiked samples. So we were happy to go forward with that. So the final method looked something like this. So filtration using a disposable filter funnel and a hand vacuum pump, then the filter paper being moved to a plastic 5mm tube with a metal ball bearing and a chemical lysis buffer and disruption of the cells through manual shaking. Then moving the sample onto further DNA extraction using this cartridge and a silica-tipped syringe, so moving through lysis buffers, wash buffers, and then a final elution buffer. And then the sample could be directly added to the PCR setup from the commercial kit, which had dried reagents, and then run on the portable PCR machine and results displayed on a mobile app. So the sensitivity of the whole test was determined using dilution series of E. coli cultures, and it was determined to be around 1,000 CFU per 100 mils of water, with detection dropping off quite rapidly after that. And the graphs at the bottom just show what you see on the mobile app, with E. coli shown by the green curves and an internal positive control by the red curves. So we then tested real samples of irrigation water sent in by end users. So they were tested with just the gold standard microbiology method to get counts of E. coli per 100 mils of water, and then also our portable real-time PCR method. Samples which contained over 1,000 CFU were detected by the method, but those over 100 CFU were only sporadically detected, and the CT values did not show a good relationship with the CFU values from the gold standard method. So finally, we had training sessions with end users, the first one which took place at Fera, where they just processed negative water samples, and then the second at an on-site location where real irrigation water samples were collected and tested. This was just to get feedback from the end users about how they felt about using the method, whether it could be useful, and see the feasibility of performing it outside of a lab environment. Just to summarize, this first method, the test showed good specificity during validation. It had a simple and equipment-free workflow that could be completed in about an hour. The feedback from end users about performing the method was generally positive. However, the sensitivity is not currently likely high enough for some of the strictest testing criteria. For example, M&S irrigation water for salad crops, you need to be seeing anything that's over 100 CFU. So it probably needs some improvements for that. And it didn't show a high correlation with the gold standard CFU counts. So again, more work would perhaps be needed to get a quantitative result. Now I'll hand over to Manisha.
Thank you, Ashley. So without further delay, I'll move to the second scenario that we selected for this pilot study. For the second scenario, we chose testing salmonella in food products. And among food products, we focused on the products which are not of animal origin, and they are of high priorities in case of biosurveillance. And among these food products, for this pilot study, we chose to test sesame seeds for salmonella using LAMP technology. So, as described by Ashley, coming to the very first part of the study was developing the method in lab, and in this phase of verification in lab, we first compared 3 LAMP assays to see which works best for us. And the assay 1 is also used by workstream 3A, 3B, and they have a poster in this conference. So all these assays were tested using a range of target salmonella and non-target bacterial strains. And so far they more or less were consistent. But among these, no cross reaction was observed. And out of these three assays, we decided to go forward with the commercial kit as it is user friendly and showed comparatively more consistent results. The next phase in validation in lab was to standardize the crude extraction method. So in crude extraction method, the idea was to develop the method as simple as we can so that it's easier for the end users to use. So in the for all these experiments, 25 gram of sesame seeds spiked with 1000 CFU of salmonella were used. And the very first step was to bring bacterial cells into the solution. And in this case, that solution was water. We shake the bottle and move to next step, which is the segregation of seeds and the solution. For segregation, we compared the cell strainer and the filter paper with funnel. So among these two, cell strainer worked best for us. And moving on to the next step, where we try to extract the cells only from the water. And here we compared the syringe filtration and filter funnel with the hand vacuum pump. And among these two, syringe filtration didn't work for us because the solution that we get from the sesame seeds was starchy in nature, therefore difficult to filter using syringe. Therefore, we decided to go ahead with the filter funnel method. In the next part, we compare two methods to lyse the cells. And here we compared heat lysis and bento DNA extraction kit. And among these two, so heat lysis is simple, easy to use, and gave us better sensitivity. Therefore, we decided to move forward with this one. And overall, the final crude extraction method looks something like this in the figure, where first we shake the bottle to bring the bacterial cell in solution, followed by segregating, filtration, and heating for lysis. After the final step, the upper clear lysate was spiked in the lamp reaction. So after standardizing the crude extraction method, next we decided to look at the sensitivity of the whole method. And for the entire verification in lab part, we used a variety of sesame seeds, that is mixed seeds, white, brown, and black, so that we can know the efficiency or performance of the test on all the varieties. So to determine the sensitivity, we spiked the samples with 10 raised to power 5 CFU to 10 raised to power 1. And the limit of detection was nearly 10 raised to power 3 CFU, where we got consistent result for all variety of seeds. For in case of black seeds, there was, due to colored extract, there was some inhibition observed. Therefore, we decided to dilute these samples and incorporated BSA in all reactions. So once we had the method from verification in lab, we moved on to testing the real sample. And to test the real sample, we first visited the end users at Felixstowe Port. And during this visit, we saw the sampling halls, interacted with the inspectors during inspection, and gained insights about how the inspection is done at the borders. And overall, this visit was really useful to gain understanding about how trade happen at the borders, and what are the logistical requirements or testing requirements that we need. Unfortunately, the samples obtained during this phase were all negative for salmonella, which is good news as a consumer. But as a molecular biologist, yeah, we would love some positive material. Anyway, so from verification in lab, we had the final method. Therefore, we decided to move forward to trialing this method in the hands of end users. And during this part, two port health officials were invited at Fera. invited at Fera, where they first saw the demonstration of whole process and followed by hands-on practice. After training at Fera, we visited Felix store and there to perform one set of supervised on-site testing, and then we set up everything, and the end users, that is port health officials in this case, they performed the testing independently for about a month or two. And in during this phase also the samples were negative, but it is really encouraging to note that the end users, they were able to independently perform the whole method by themselves and shared us the results. And the samples that they tested on site were also tested at Fera to confirm the results. So to summarize, the specificity from the lab verification part, the specificity of the test was good, and so far, because there are some experiments that is still ongoing. And in terms of usability of the test, the feedback from end users was really positive. And in terms of sensitivity, it could use some improvements, as the requirement is no salmonella in 25 gram of seeds. There is a challenge in developing the crude extraction method in journal for food matrices, and the challenge we observed in case of sesame seeds as well. And in terms of overall approximate cost of this test per sample was around 20 to 25 GBP, plus one-time instrument cost, and the time to result was around 1 hour. With that, I would like to summarize the outcomes from the entire work stream 3A, which was divided into 3 work packages. So in work package 1, a broad literature survey was conducted to see what technology is out there. And in this literature survey, more than 16,000 papers were screened for their title and abstracts. Moving further, nearly 3,000 papers were screen for full text review. After that, we delivered a technology readiness level framework, and combining these 2 points, the database of the technology and their readiness level was determined in this work package. In work package 2, interviews were conducted with stakeholders and end users, and this engagement from this engagement, opportunities around saturatory and non-saturatory testing were determined. So for work package 1 and 2, we also have a poster if you want to know more about them. This talk was mainly focused on work package 3, and in work package 3, based on the information from work package 1 and 2, 2 technology and scenarios were selected for pilot in-field diagnostic testing. And from validation point of view, both assays showed good specificity, and and real samples were also tested. In the last phase, the end users were trained at Fera, followed by testing on site, and their feedback was also gathered. Like with any trial experiments with the technology, there were some issues, or we can say lessons learned during this study, where we can focus more in future. So I discussed some of the technical aspects in my previous slide around challenges in developing crude extraction and sensitivity. Another challenge could be interpretation and troubleshooting of results, where you need a trained laboratory personnel. Coming to the logistical issues, to perform any on-site testing, the requirements around infrastructure, staff, and appropriate facility is needed. Another one is to maintain the proficiency and quality of the testing. Regular trainings are required. Lastly, to make the decisions based on the on-site testing results, some changes in the legislation could also be discussed further. With that, I would like to thank everyone who contributed to this project, and acknowledge PathSafe program members and members of Fera that took part in this study, and our collaborators from University of Lincoln, our end users who we really appreciate their contribution in this study, and CFAS for giving us DNA for specificity experiments. Thank you.
Thank you very much. So we move from work stream 3 on remote diagnostics to work stream 4 on environmental AMR, and I can see someone moving to the stage now who's definitely not Alwyn Hart. So Webke, over to you.
Please. Thank you. Hi, everyone. My name is Wieke Schmidt. I'm not Alwyn Hart, so apologies for everyone who came to see and speak to Alwyn today, but I'm working with Alwyn in the Chief Scientist Group at the Environment Agency. And yeah, today we're going to present to you what we have been up to in the work stream 4. So to kick off, let's remind ourselves where we started, because sometimes it's easy to forget. And that is, we had the National Action Plan, which comes to an end now, which had a specific commitment to explore environmental surveillance for AMR. And this is in line with other international calls on organizations, such as from the World Health Organization and the United Nations Environment Programme. So when we look at guidelines for surveillance, such as the WHO Global Tricycle Surveillance, and translate that into the environmental realm, then this looks a little bit like this schematic here, because they kind of recommend to use an indicator organism that could be monitored in clinical, veterinary, and environmental settings, such as ESBL coli. We know some of the sources, such as wastewater treatments, animal source slurries, and we heard quite a lot today and yesterday about it. And we know some of the potential pathways could be through our waterways for transmission towards people or animals as receptors to this hazards. So while this is a start, however, there are some missing linkages. For example, we know that antimicrobial resistance encompasses not just antibacterial, but antifungal, antiparasitic, and antiviral. So if we take this concept of hazard, source pathway, and receptor further, our choices for an environmental surveillance should consider the wider term of AMR. Here we have included how the concept could be applied to antifungal resistance. For example, we know Aspergillus can develop resistance to azole fungicides, which are applied to crops, and that resistant spores and fungi can be transported through the atmosphere to people. So in other words, if we think of the range of potential risk linkages, then we end up with more something like this schematic overview. And coming back to environmental surveillance, the choice of which pathways or in what environmental surveillance takes place should be determined by what risk we wish to understand, and perhaps also by what and where we can mitigate. But of course, it will be unavoidable that a surveillance program cannot cover every of these source path receptor linkages. So in workstream 4, we have conducted a wide range of projects to address the NAP commitment, specifically to identify appropriate methods that are suitable for monitoring of a range of resistant microbes, genes, and antimicrobial substances in the air, water, and soil. Secondly, we tested this method in the field, as many of our challenges that we expected to face were the practical ones, such as logistics, cost, and resources, and we just heard a good example in the earlier talk. Thirdly, we also aimed to increase our understanding of the selection pressures of antimicrobials, and the workstream also developed a data system, and that has been introduced by Oliver yesterday. So I'm not going to go and cover this because he excellently explained this already. So in a nutshell, we really wanted to explore what an environmental surveillance can provide and equally what it cannot provide. As I mentioned, we conducted a wide range of processes over the last 18 months. And as you can see, we have done this with a range of partners. The works ranged from, from surveillance around the air, the water, but we also looked at antifungals and biosolids. We were interested in selective pressures for antimicrobials, and we took first steps towards a risk assessment. So over the next slides, I'm going to give you a short overview about our work. And to kick off with this is our river pilot work, where we sampled from May 2022 to February 2023 surface waters at 3 river catchments. These catchments were selected as they represent different circumstances, such as urban settings and rural settings, and they were impacted by different land use. We aim to explore these geographical differences between and within the catchments, and you can see more information about this work by Katie's poster in the foyer. It will be the first on your right, I believe. So from the water to the land, where we tested for the presence of 14 clinical antifungals in biosolids. This work used samples from 11 sludge treatment centres in England and Wales, which were sampled within the Chemicals Investigation Programme. The work has been published in the Science of the Total Environment, but just 2 kind of key findings. One is that ketoconazole and micozole were detected in all of the samples. And secondly, that the levels of itraconazole were 2 orders of magnitude higher than the indicated predicted no effect concentration for resistance in soil. And it's quite a mouthful. But it leads me to our work on selective pressures. There are not many threshold values available that inform at which concentration an antimicrobial substance there are not many threshold values available that inform at which concentration of an antimicrobial substance resistant microbes have an advantage to non-resistant microbes. So if some of these values exist, they are mainly for antibiotics. And across 4 projects, we aim to get a better understanding at these values for selective pressures of antimicrobials. And for example, one of these projects explored concentrations of antifungals, where we compared competitive advantages to resistant yeast strains in comparison to non-resistant strains. Overall, the outcomes of all of these projects help us to bring detected environmental concentrations into context of what it means for resistance. This work included not just antibiotics or antibacterials and antifungals, but also disinfectants. Another strand of our work included the evaluation of existing literature and surveillance efforts to explore the potential use of sentinels as bioindicators for future environmental AMR surveillance. The literature review revealed that the most targeted host taxa for AMR investigations were birds and mammals, and very few studies reports were found on plant species. Secondly, there are few current surveillances, with 3 schemes identified that currently collect AMR data whilst performing pathogen-focused research. Filter feeding shellfish presents a potential useful sentinel bioindicator model for marine waters, and we collaborated with CFAS and to explore this further. And Craig has a poster also out in the foyer. I think it's also on the right side, so you can learn more about that. To date, much of AMR in the environment has been conducted in the aquatic or in the terrestrial realm, and the atmospheric microbiome has often been neglected. Yet, as I mentioned at the start of this presentation, we know that some of the resistant microbes can be transported through the atmosphere. So we reviewed available sampling options for atmospheric antimicrobial resistance, as well as exploring the prevalence of it near potential sources. Overall, our work has led us to develop or taking the first steps towards a risk assessment. In one of our last projects, we have developed an approach that aims to assess a range of the environmental scenarios for human exposure to microbes resistant to antimicrobials, and thus compare the significance to human health. So to summarize, we have been really busy over the last 18 months, and our work aimed to identify and test the hazards and drivers for AMR in environmental settings. We also investigated on how we can define threshold concentrations and taking first steps towards a risk assessment. The majority of our work has already been published, and you can most of the findings on gov.uk. A few more reports are coming this spring, we promise. It's on high priority to get it out there, and so we can share it with you. And while the PathSafe funding for us has finished, our AMR in the environment work has not. We have currently ongoing work where we explore further the fate of resistant microbes in an urban river. We also continue to looking at the development of antifungal resistance. So again, there's more to come. But for now, I would like to thank all my colleagues at the Chief Scientist Group, at the Environment Agency, but also about our partners at UKHSA and other partners. And yeah, thank you for listening, and apologize, I'm not Alvin. So.